आज का हमारा लेक्चर है रेड एल्जी। रेड एल्जी का मेंबर है पोलिसिफोनिया। रेड एल्जी की डिवीजन होती है रोडोफाइटा और इसकी फैमिली होती है रोडोमाइलेसी। पोलिसिफोनिया is a marine alga commonly found along the coast of the oceans. It is attached to the rocks or the epiphytes. जैसा कि मैंने आपको पहले लेक्चर में भी बताया था, एपीफाइट्स का मतलब क्या होता है कि ये अलगल स्पीशीज जो है ये दूसरी अलजी के ऊपर जो है ग्रो करती हैं या किसी प्लांट के ऊपर ग्रो करती हैं। The vegetative structure of the plant body is branched, filamentous, and basal attachment disc. It has rhizoids. Rhizoids, these are the root-like structures. The basal attachment disc is formed of the several non-septate branch rhizoids. ये रूट्स एक्चुअल नहीं होती बल्कि ये रूट लाइक स्ट्रक्चर्स होती हैं, इनमें वास्कुलर बंडल नहीं मौजूद होता, जबकि रूट्स के अंदर वास्कुलर बंडल्स मौजूद होता है। ये डिफ्यूजन प्रेशर के जरिए या ऑस्मोटिक प्रेशर के जरिए वाटर को अब्सॉर्ब करती हैं। वेजिटेटिव स्ट्रक्चर में इसके जो है ना वो इसका इसे कुछ पोर्शंस होती हैं, जिसमें फर्स्ट पोर्शन जो है इसका इरेक्ट पोर्शन कहलाता है। The vertical portion is formed of laterally branched axis, which is polysiphonous. Its axis is formed of a bundle of filaments or the siphons. The cells of each filament lie at the same level. These cells are placed one above the other and form the bundles of siphons. The central siphon is primary axis. It is surrounded by variable number of the pericentral filaments. The number of pericentral filaments in each style is constant for a particular species. ये इरेक्ट पोर्शन जो है उसकी ये कैरेक्टरिस्टिक्स होती हैं। इसमें एक सिफन होता है जो फिलामेंट्स को सिफन कहते हैं। सिफन इंग्लिश में आमतौर पे छलनी को कहते हैं, जिसमें बारीक बारीक से सुराख होते हैं। Colticating filament. The central filament is further surrounded by one or more layers of the colticating filaments. उसके बाद है ट्रैकोब्लास्ट. The fine hair-like structures called the trachoblasts, because at the tips branches are the monosiphons. The cells are uninucleate or the colorless. उसके बाद वेजिटेटिव स्ट्रक्चर के बाद हम इसके सेल स्ट्रक्चर को स्टडी करेंगे कि इसके सेल के अंदर क्या क्या चीजें मौजूद होती हैं। सबसे पहले उसकी सेल वॉल होती है। इट इज मेड अप ऑफ द सेलुलोज एंड द पेक्टिन। जैसा कि पहले वाली प्लांट्स अलगल स्पीशीज के अंदर भी जो है ना वो पेक्टिन बड़ी इंपॉर्टेंट है। जो सेल वॉल का जो हिस्सा है, सेलुलोज तो तकरीबन सबके अंदर मौजूद है, लेकिन किसी के अंदर पेक्टिन मौजूद है, किसी के अंदर कोई और मटेरियल मौजूद है। क्लोरोप्लास्ट are discoid in shape. The pigments are the chlorophyll A and carotene xanthophyll. उसके बाद है जी फैकोरिथ्रिन and फैकोसायनिन. These are the pigments which are generally present in the Rhodophyta or the Polysiphonia. It lacks perinoid. The reserve food material is fluoridine starch. Starch, you know that. What is the starch? Starch is a polysaccharide. Young cells are always uninucleate, but the older cells are the multinucleate. शुरू शुरू में जो सेल्स डिवाइड होती हैं उसमें एक सेल होता है, और जैसे जैसे ये सेल बड़े होते चले जाते हैं उसमें नंबर ऑफ न्यूक्लियस की जो तादाद होती है वो बढ़ती चली जाती है। This is the vegetative structure of the Polysiphonia. You can see that this is the pericentral siphon. This is the old polycyphorous long branch. उसके बाद है जी this is the nucleus, this is the chromatophore, this is the transverse section of the pericentral portion of the siphon body. This is the pericentral cell, this is the axial or the central cell. The other structure is the apical cell, from where the new cells produces the new filament or the new thallus-like structures. These segments, branch initials, young long branch or trachoblast are the part of the thallic-like structures of Polysiphonia. The reproduction in Polysiphonia shows an asymmetric alternation of generation. Both gametophytic, haploid and the diploid generations are identical. इसमें गेमेटोफिटिक जनरेशन जो होती है ये हैप्लॉइड होती है और स्पोरोफिटिक जनरेशन जो है ये डिप्लॉइड होती है. उसके बाद सेक्सुअल रिप्रोडक्शन में मेजॉरिटी ऑफ द स्पीशीज ऑफ हिट्रोथैलिक. हिट्रोथैलिक का मतलब होता है डायोशियस मेल एंड फीमेल सेक्स ऑर्गन्स ऑन द डिफरेंट प्लांट्स, यानी कि डिफरेंट उसके थैलस के ऊपर मेल सेक्स ऑर्गन एक प्लांट के ऊपर होता है और फीमेल सेक्स ऑर्गन दूसरे प्लांट के ऊपर होता है. इस कंडीशन को हिट्रोथैलिक कहते हैं या डायोशियस दूसरे हायर प्लांट्स के अंदर कहा जाता है. अंडरस्टैंडिंग के लिए डायोशियस लिखा जाता है ताकि हेट्रोथैलिक का जो है ना कंसेप्ट जो है ना वो समझ में आ जाए. Spermatangia. There are two types of the sex organs are produced in the Polysiphonia. Number one, the male sex organ that is known as the spermatangia. These are produced in clusters on fertile branches. The pericentral cell of the branch consists of one or more spermatangial mother cells. Each spermatangial mother cell produces one or four spermatangia. The contents of the spermatangium are transformed to single or non-motile spermatangium. This is the basically reproduction or the formation of the spermatangia or the male sex organs. The spermatangium is released on the rupturing the spermatangial wall. Once the spermatangial walls are the spermatangia matures, the spermatangial wall burst or ruptured to release the spermatangia. The female sex organ is known as the carpogonia. They are produced singly on the female fertile branches. The apical cell of the carpogonial filament transforms into a carpogonium. Each carpogonium is flask-shaped with basal swollen portion. Egg is present in swollen portion. Upper neck-like portion is called the trachogyne. After the production or the formation or the maturation of the thallus-like structures of the Polysiphonia or the formation of the male and female sex organs, the fertilization occurred. In fertilization, the spermatangia या spermatium falls on the trachogyne. ये स्ट्रक्चर्स में आप देख सकते हैं. दिस इज द ट्रैकोगाइन. ट्रैकोगाइन जो है जस्ट लाइक दैट ऑन द स्टिग्मा ऑफ द हायर प्लांट्स. जैसे फ्लावर के अंदर स्टिग्मा होता है उसके ऊपर पोलन ग्रेन्स गिरती हैं तो वो उसमें पोलन ट्यूब बनती है. वो पोलन ट्यूब जाके फिर एग को फर्टिलाइज करती है. Just like that of the trachogyne, in thallus-like structure of the Polysiphonia, the spermatia are falls on this portion. Then the spermatia germinate and goes into the carpogonium and form the zygote. The spermatial cells enters into the trachogyne and moves down to the basal portion of the carpogonium. Then fertilization occurred, form the zygote nucleus. It soon divides into two nuclei. एक zygote एक nucleus होता है और जल्द ही इसमें दो nucleus बन जाते हैं. After fertilization, the supporting cell cuts off the auxiliary cells. Auxiliary cell, supporting cell and cells of sterile filaments are together. They form a single large cell called the placental cells. This is the basal sterile filament. This is the auxiliary cell. यहाँ से हम देखते हैं A, B, C, D and E. These are the structures from where we can easily understand how the fertilization occurred. This is the supporting cell. The supporting cell then further develop into the carpogonial mother cell. Then carpogonial mother cell further develop to produce the trachogyne, or the basal portion is the carpogonium. This is the carpogonial filament. The carpogonial filament produces the trachogyne at the maturation stage. Then the spermatia fertilize to the carpogonium and produce the zygote. Several small protuberances are produced on the surface of the placental cell. The tube is cut off by formation of the septum. It functions as the initial of the gonimoblast filament. This initial undergoes few transverse sections and produces the gonimoblast filament. Then gonimoblast filament is produced, which is basically an isolated structures. Then isolated structures further develop to form the mature thallus. The terminal cell of each gominoblast filament acts as the carpoborium sporangium. It produces single non-motile diploid carpospores. The mass of the gominoblast filaments and the placental cells, the carposporophyte. It is covered with a sterile covering the pericarp. Pericarp, just like that of the higher plants, once the fertilization occurs, zygote is formed. After the formation of the zygote, it is covered by specific type of the layers that is known as the pericarp. Just like that of in the Polysiphonia, the pericarp is formed and covered the zygote or the carposporophyte. The pericarp forms of fruiting body are the cystocarp. अब पोलिसफोनिया के अंदर जो फ्रूटिंग बॉडी होती है उसको सिस्टोकार्प कहते हैं, ये आपने याद रखना है. The carposporangium rupture and release the carpospores through the ostiole. Ostiole, this is a very minute pore through the carpo through the trachogyne, just like that of the trachogyne or the micropyle. The carpospores germinate to produce the sporophyte. Sporophyte is also known as the tetrasporophyte. It produces the tetraspores. Asexual reproduction. Asexual reproduction takes place by the production of the tetraspores. The pericentral cell itself divides transversely to form the two unequal cells. The lower smaller stalk cell, the upper largest cells, tetrasporangium. The nucleus of the tetrasporangium divides by meiosis to produce the four haploid tetraspores. It means that the tetraspores are the haploid cells. The sporangium ruptures and spores are released. The haploid tetraspore germinate to form the gametophyte. This is a schematic representation of the formation of the tetraspore and the formation of the zygote, and the zygote produces the tetraspores. From here, the tetraspores, which are the haploid, the male and female plant produces the spermatangia and the carpogonia. The carpogonia spermatangia fuse to form the zygote, and then it produces the tetraspores. This diagram shows the life cycle of the Polysiphonia. This is a male gametophyte, this is a female gametophyte. Male gametophyte produces the spermatia, female gametophyte produces the carpogonia. The spermatium and carpogonium, when fused to produce the when fertilized to produce the auxiliary cells. The auxiliary cells further develop into the basal sterile cells with the help of the sporting cells or the carpogonial cells. Then this carpogonial cells further develop into the large carposporangia. Then carposporangia produces the carpospores. The carpospores, which is the 2N, and further germinate to produces the 4 cells. These are known as the germinating carpospores. Then carpospores, which is the 2N, further develop into the 4 tetrasporangiophyte. This is the tetrasporangiophyte. In tetrasporangiophyte, further meiosis occurred, produces the tetraspores. Each sporangium produces the 4 spores, which are the haploid. Then haploid tetraspores produces male gametophyte and the female gametophyte. In this process, the Polysiphonia life cycle occurs.